This free presentation is brought to you by Quantum University.
Hi, dear friends, cultural creatives, and healers everywhere. Thank you so much for joining me in today's presentation on the nature of stress and its influence on telomeres. As we read the papers every day and watch the news, what we're beginning to find is that there are crises affecting the entire planet in every form. One of the most important of these crises is the healthcare crisis. When we try to understand why are we facing so many crises, it's because we're undergoing an evolutionary upheaval on our planet. And the planet is facing tremendous problems. And how are we going to get out of it? Well, an important insight into the solution is provided by Albert Einstein, who said, "We can't solve the problems with the same kind of thinking we used when we created them." What Einstein is getting at is that the crises we face today are actually of our own making. And we really have to understand what is it that is causing these crises. Well, as Einstein mentioned, it's the way we're thinking. We really changed our beliefs around 1953 when we started to look at health as a consequence of genetics. It was in 1953 that Watson and Crick provided the world with the understanding of a DNA double helix, and that the DNA is a blueprint to provide for the body's proteins. The significance is this: the character of our lives, the behavior we express, and the nature of our health is all tied up with the proteins in our body, because the proteins give us our structure and give us our behavior. So when Watson and Crick revealed that the source of these proteins were the blueprints called DNA, all of a sudden science started to recognize that the fate that we express in this world, the fate in regard to what we will be able to accomplish and our health, is all tied into the genes that we received at conception. So the significance is when we look at today's model of health in the medical arena, it is recognized that the body is a biochemical machine controlled by genes. The significance of this model, which really reflects the nature of something we call genetic determinism, is illustrated in the cover of this Life magazine. "Were you born that way?" says the title. And what is meant by this is the issues involving our physiology, our behavior, and our emotions seem to be derived from the genes that we received at conception. The significance, again, is that our understanding of the nature that genes are controlling these proteins and that the proteins provide for those issues of our physiology and health leads us back to saying that the genes are the cause of our issues. In fact, when I was teaching in the medical school over 30 years ago, the primary message we provided to the students was a concept called genetic determinism. Genetic determinism is the belief that the genes are self-actualizing, meaning that genes turn on and off. Then they produce the proteins that create the characteristics of our lives. So in reverse engineering the concept of health crises, I say, well, what would be the cause of it? And as we back up, we say, well, it's something in the physiology of the body, which then takes us back to the proteins, and then finally you say, yeah, but where did those proteins come from? And here we are at the DNA. The concept of genetic determinism, unfortunately, is a belief that we are victims of our heredity, that we didn't pick the genes that we came with as far as we know. We don't like the characteristics the genes provide. Well, we can't change them. And then we recognize from the work of Watson and Crick that the genes are self-actualizing and turning on and off. And you put all that together and you realize, my God, our health issues are not in our hands, that we are the victims of the genetics, the heredity that is running in our family. This idea that genes control life is actually then put into the nature of cell biology in regard to where is the brain of the cell, because that's where the control comes from. And if we look into the textbooks of today, we find that the nucleus of the cell is given the credit as being the cell's brain. The reason why the nucleus was selected is very simple. The nucleus is a repository of up to 98% of the DNA in the cell. With the belief that the DNA was self-actualizing, then it came down to the fact that the gene programs in the nucleus were automatically engaging, and through that, we express our life. The point is, since we have no control over the genes, and the genes control us, then we perceive of our issues in life as we are victims. And therefore, we seek a rescuer to try and help us understand how we can then return back to health. Well, the idea that the nucleus is the brain of the cell has been in our understanding since Watson and Crick, and actually back in the late 1950s. And the question is this actually a true statement? Well, in that regard, we can look at the issue as this way. If you remove the brain from an organism, the consequence is that the organism will die. Then I say that if we go to the cell and remove the nucleus, the brain of the cell, then the cell should die. But the issue is, in the process of enucleation, that is, removing the nucleus of the cell, the result doesn't cause the cell to die. As a matter of fact, the cells can survive for weeks, if not months, without any genes at all. And without this genetic material, the cells are still capable of controlling their lives, even without the DNA. So the issue is, where in the cell are we going to find the brain? Because obviously, enucleation reveals that the brain is not controlling the day-to-day functions of our biology. So, in search of the brain, we're going to have to look into another aspect of cell biology and cell function. I was actually introduced into the nature of what controls the cells through my research on cloning stem cells back in 1967. Stem cells are actually embryonic cells. As a matter of fact, moments before you were born, if I took a biopsy and showed you a cell in a tissue section, and I say, this is an embryonic cell, but then did the same observation a moment after you're born and found the exact same cell, no longer would I call it an embryonic cell, because now that you're born, we change the name, we call it a stem cell. Question might be, why do we have embryonic or stem cells in our body? And the answer is quite simple, is because every day we lose hundreds of billions of cells from our biology. And you must remember, the biology that we express is the result of 50 trillion cells coming together in a community creating the human body. If we are losing hundreds of billions of cells every day, then there's a very serious issue. If you do not replace these cells, then we will not be able to sustain our life, because we'll lose too many of the cells that control for our biology. The conclusion is simple. Stem cells are present in the body to keep us alive, because their function is to replace the cells that die from normal attrition every day. So, I was cloning stem cells, these multipotential cells, and that means I would just put one cell in a petri dish by itself. The stem cells divide every 10 to 12 hours in a tissue culture. So, at day one, I put one cell in, and 10 hours later there are 2 cells, and then 10 hours after that there are 4 cells, and 10 hours after that there are 8 cells, and what you can see is every 10 hours the cell population is doubling. After a week of doubling, I end up with about 25 to 30,000 cells in the petri dish. Now, the most important insight into the experiment so far is that all 25 to 30,000 cells are genetically identical, because they were all derived from one parent cell. So now that I have genetically identical cells in the dish, I split the population into 3 separate petri dishes. So each dish has genetically identical cells in it. But the difference in my experiment comes in when I feed the cells with culture medium. Culture medium is the environment in which cells grow. I say, well, what does culture medium represent? I say, culture medium represents the blood of the organism from where I get the cells, because the blood is the environment for those cells. So if I'm growing human cells, I look at the composition of human blood, and then in the laboratory create a synthetic version of that blood. If I'm growing mouse cells, on the other hand, then I look at the composition of mouse blood and create culture medium based on that chemistry. So I am creating culture medium for my cells. Because I'm synthesizing the culture medium, I can actually change the composition. And in changing the composition slightly in each of 3 different versions, I create 3 different culture environments for the cells fed with that culture medium. So in culture medium A, when I feed cells with that variation, the cells form muscle. In the second dish, where I feed them a second variation on the culture medium, environment B, the cells form bone. And in the third dish, Again, with a variation on the culture medium, environment C, the cells form fat cells. So the results are very profound because it says the cells in dish A form muscle, in dish B form bone, and in dish C form fat cells. And the question is, well, what accounts for the fate or differentiation of these cells? Well, number one, all the cells were genetically identical. So any evolution after that is not due to the genes, but it turns out it's due to the environment in which the cells are living. The composition of that culture medium determines the fate of the cells. Well, this is really profoundly important for a very simple reason, is because it says that rather than genes controlling the fate of the cell, it's the environment in which the cells live that determine the fate of the cell. In the big picture, this is very important because in the old story where genes control us, we become victims of genetic programs that are in the nuclei of our cells. But the new insights provided from these stem cell cultures reveal no, it's not the activity of the genes that determine the fate, it's the situation in the environment that is influencing the fate of the cells. And when you understand that, then all of a sudden you understand a very profound insight. And that is, we, by being able to change the environment, are the ones that control the activity of the cells. Well, this science that I saw in 1967 is now the leading edge of research in conventional biomedical studies, and it's really under the title of something called epigenetics. That was the name that was provided to the observations of environment-controlling genes back in 1990. So we have a new science, epigenetics, and you say, well, that sounds just like genetics. I go, it is a profound revolution for this. Old belief says the genes control our lives, and as we discussed, then we become victims of our heredity. But epigenetics says the environment controls our lives, and all of a sudden, rather than victims, we can perceive ourselves as masters because we're the ones that can control the environment. So it becomes a major revolution in biological insight, a new way of thinking of the issues in regard to healthcare and the crises that we're facing now. And that is this: we're not victims. We are masters, and as a result, if we understood that, we would be able to control our health and not have to deal with the crises in healthcare that we face right now. So we're left with a very important biological fact, and that fact is simply this: genes do not control biology. Now, this is a belief that almost everyone has been programmed with, and even though leading-edge science no longer entertains that belief, the public is still endowed with the fact that their heredity is influencing the expression of their life today. Well, the big problem is this: it comes down to a confusion between two terms. The two terms are correlation and causation. The significance is this: causation means the act or agency which produces an effect. Something causes this. Correlation simply means a relationship exists between a phenomena and other things. So, for example, the old belief, genes cause disease, meaning genes are responsible for disease, is totally incorrect. Because as we recognize in epigenetics, genes are not controlling the expression of life. A more appropriate term is that genes are correlated with diseases. Now, this is profoundly important because it emphasizes the fact that the genes are actually not the cause of the problem. Correlation, on the other hand, is different than causation because correlation just means that there's a relationship existing between phenomena. So it is true, genes are correlated with disease, but genes do not cause disease. The new insights of epigenetics reveal that the environment in which the organism lives is responsible for shaping the genetics. So this accounts for a very important insight. For example, so many people believe that breast cancer is associated with a gene called the BRCA1 gene. And when women hear they have this gene in their genome, most of them get very upset because they automatically think, oh my God, I have the breast cancer gene and I am ultimately going to be a victim of that breast cancer. Well, there's something to consider that's profound here, and that is this: only 50% of the women that have this gene actually get the breast cancer. What's the point? Well, 50% of the women have the gene and never get the breast cancer. So A, does the gene cause cancer? Answer, no. Why? 50% of the women having the gene never get the cancer. B, though, is the gene correlated with cancer? Is yes. But that doesn't mean it causes the cancer. So you might have to say now, what is the difference between the women that get the cancer with the gene and the women that have the gene and don't get the cancer? And the answer comes down to a very simple insight, and that is the behavior and response of an individual to their environment via epigenetics is causing the disease. Then you stop long enough and say, well, wait a minute. If I can change my perceptions and I can change my environment, I am then not a victim of my genes. I am the master because I am controlling the situation that in turn controls the genes. So we have to let go of the old belief system that is actually relating to the healthcare problems, because when you tell a population that their issues are related to the genes and they have no opportunity to influence those genes, then the belief is people see themselves as victims, and that is really where the cause of the disease comes from, that belief. So I say, okay, we have an opportunity to review the idea and see what is different in today's world. As Albert Einstein said, let's think different from the way we did that caused the problems. And epigenetics is the new thinking. So I say, okay, let's start from scratch. I say, well, how do cells work? Well, the first thing is this. Cells are comprised of protein building blocks. There's a family of about 100,000 different proteins that, when assembled into different configurations, are responsible for the cells that make up our body. So consider the proteins as a giant Lego set that you can take 100,000 different parts and assemble them in different configurations, and as a result you can create brain cells, nerve cells, muscle cells, skin cells, all using the same different building blocks but in a different assembly. So, number one, proteins provide for our structure, and then all of a sudden we say, oh my goodness, if you understand the nature of the proteins, guess what? You now have an understanding of the secret of life. And the secret of life is actually so simple, because the secret of life is movement. Meaning, if something moves, it's pretty much alive, and if something doesn't move, it's not alive. I say, yeah, but where does movement come from? And this is the most wonderful insight. Movement is derived from the proteins. Yes, the proteins have structures that provide for the body, but the most important insight about the nature of proteins is that they're capable of interacting with environmental signals. So when an environmental signal is introduced to a protein, and that signal is in resonance with that protein, in harmony with that protein, then that signal can cause the protein to change its shape. And we call shape in biology conformation. So a signal in the environment can cause a protein to shift from conformation A to conformation B. But as you can see in this video, as the proteins shift from A to B, it causes the proteins to move. And I go, exactly, that's the source of life. The movement of shifting proteins is harnessed by the cell to do work. Work requires movement, and the movement comes from proteins that are shifting from one conformation into another. To give you an example of that, let's consider a protein called kinesin. Kinesin is a protein that looks like two pipe cleaners that are twisted together in a helix, and at the bottom of each of the pipe cleaners is a globular protein that resembles a foot. I say, well, what's the relevance about it? And I say, well, when the kinesin molecule touches a signal, in this case a microtubule, the kinesin is forced to change its shape. The significance, as you can see in this video, is in one conformation, the left foot or globular protein is the leading foot in the movement. But when it touches the microtubule, it causes the protein to change its shape, and that causes the right foot to move in front of the left foot. And then when the right foot touches the microtubule, again it causes a shape change, and this time it causes the left foot to come in front of the right foot. The result is simple. the kinesin, upon touching the microtubule signal, starts moving from right foot to left foot to right foot to left foot and is essentially walking down the microtubule. To give you an idea of what that really represents in the cell, the next video shows a microtubule and the kinesin molecule. Well, the kinesin molecule represents the protein that we're talking about, and the microtubule is the substrate or the signal provider that causes the kinesin to change shape. This particular kinesin, the pipe cleaner portion of the molecule, is connected to a vesicle in the cell. So as you can see in this dynamic picture, as the kinesin is moving along the microtubule, walking right foot, left foot, right foot, left foot, it is carrying a vesicle through the cell to another destination. And all of a sudden you can start to see how the movement of a molecule is engaged in the expression of behavior. Well, just those little movements create life? Well, I have an interesting video right here to show you, and it's a video from the Tesla motor plant in California. Now, the Tesla, one of the most wonderful cars introduced on this planet, the electric Tesla, is built in one of the most modern factories in the world. And here you can see the factory floor. And in this picture, there are giant red machines. The giant red machines are the equivalent of proteins in this regard. The machines have different shapes or conformations. In one shape, the machine may be over here, and then when the signal occurs, the machine will move over here. And I say, yeah, but if you put these machines together in a sequence like an assembly line, then you get this picture. And as you see in this amazing video, the machines are cooperating in a pathway, one machine influencing the product and the next machine altering that same product and the next one. So as you go down the assembly line, proteins are continuously modifying the product and creating a car. And what's most interesting in this video is there are no people on this factory floor. And I say, well, then here's the beautiful analogy. The protein machines in the Tesla plant, each providing a small movement, collectively can build a car. I say, take that image, reduce it down to a nano size, down to the size where those red machines are actually the size of proteins. Consider those red machine proteins in that nano image, and I say what? The movement of the proteins, rather than assembling a car, are actually assembling the necessary building blocks of the body and creating the functions of the cell. So, how do proteins work? Exactly the same as the machines in this Tesla plant. Well, then we can now come to a very important conclusion about proteins. Proteins are the building block of the body. But in the presence of an environmental signal, the protein can change its shape and form a different conformation. The change of shape generates movement, which we might refer to as behavior. So I say, where does the behavior of biological organisms come from? And the answer is, when signals complement the protein building blocks of the body, causing them to move, that movement is then creating all the characteristics of life. Well, we know where the proteins come from. The proteins are the product of the DNA blueprints. The question is the signal. Where or what does the signal represent? And why that's becoming relevant is by tracing the signals, we then can understand the nature of the biology of the cells. So, in this illustration of a cartoon version of a cell in a petri dish, it shows that when I add the culture medium with environmental signals that I create in the laboratory, and I add that culture medium to the cells, and you can see the chemicals coming down, and some of them start to stick on the surface of the cell. This environmental signal then alters the behavior of the cell. And how does it do it? And the answer is simple. When the signal hits the surface of the cell, the skin or the membrane of the cell, the membrane sends a complementary signal inside the cell, and that second message, and that's what it's called, that second messenger is the signal that activates the proteins of behavior. So an environmental signal can be picked up by the membrane of the cell, relayed as a secondary signal into the cell, and that secondary signal controls cell behavior, and as we also find out, it can control the genetics. So the whole process of signals controlling biology is called signal transduction. But the special part where the signal actually is influencing the genetic readout is called epigenetics. And that is how the signal controls our biology. So what we really want to know is, how is the cell controlled? And the answer is the membrane. The membrane is how the cell is controlled. The skin of the cell is the brain that manipulates the signals that control the expression of the cell. And you go, wow, the cell has a brain and it's related to the skin. And I go, interesting, because when you understand human embryology, I say, where did the human brain evolve from the embryo? And the answer is the skin. The skin of the embryo is the source of the brain. So the cell and a human are exactly the same in regard to this, is that both of them represent the brain that read environmental signals and then adjust the behavior of the cell. So I say, well, how does an environmental signal select the genes? So I say, when you open up the nucleus and remove the hereditary units called chromosomes, this is the unit that actually provides for biological characteristics and heredity. I say, yeah, a chromosome is a hereditary unit. Yeah, but a chromosome is made out of 50% protein and 50% DNA. Yes, a chromosome is made out of 50% DNA and 50% protein. Well, since Watson and Crick in 1953, science was so interested in the DNA that when it would isolate the chromosome, it would separate the DNA from the protein, save the DNA for the research, and then throw away the protein. At some point, somebody finally said, listen, half the nucleus is protein. What is that protein doing? Now we have a new insight, and that is this. A chromosome represents a core of DNA surrounded by a sleeve of protein. And the significance is, when an environmental signal complements the proteins on the DNA, it causes the proteins to change their shape. And when the proteins change their shape on the chromosome, they pop off of the DNA molecule, exposing the DNA blueprint. Then other proteins come up, copy that DNA blueprint into a Xerox version of the gene called RNA. That RNA molecule goes into the cell and is decoded in creating a protein. So we have an insight. How does a gene work? And the answer is this. A gene is a blueprint. It has no self-actualization. The gene makes no decisions at all because the activity of the gene is controlled by the environmental signals, which in turn control the protein sleeve on the DNA. So we've been focusing on, DNA controlling life. The epigenetic story actually shifts our focus to looking at how environmental signals are controlling the DNA in our lives. So all of a sudden, it changes our belief system. It says, no, DNA is not self-actualizing. DNA expression is related to our response to the environment. So I say, okay, let's go back to the cells in tissue culture. And as I said, as I put the tissue culture cells in the plate and then feed them with culture medium, it is the culture medium that determines the fate of the cells. And I go, well, here's an interesting point. Well, we've been talking about cells in a petri dish. It's really no different than cells in your body. Remember, your body is comprised of 50 trillion cells. That, in fact, you can perceive of your body as a skin-covered petri dish, and within that dish are 50 trillion cells. So a human body is a petri dish. And I say, yeah. And just like our study in the laboratory, adding culture medium to the cells, well, in the body, the culture medium is the blood. So all of a sudden we say, well, it is no difference if the cell is in the plastic dish or the cell is in the skin-covered dish, because the cell is still going to respond to the culture medium environment. And in the human body, that culture medium is blood. I say, well, that becomes important because then the chemical composition of the blood is actually controlling the fate of the cells. And I say, well, there are two aspects to what is in the blood. that is controlling biology. Number one is just the basic nutritional elements. So in culture medium, there's nutrition. And this is significant because it says when we make the culture medium called blood in our human body, it also is derived from nutrition. So one of the big factors affecting our health is, are we taking in proper nutrition that will support the health of our bodies, or are we taking in toxic elements as produced in industrial farming? Because these toxic elements will interfere with the growth medium, and in turn, via epigenetics, the toxic food that we eat will alter our genetic expression and can contribute to our health. Well, in addition to nutrients, blood contains cellular signals. I go, cellular signals? I say, yeah. Emotional chemistry, hormones, growth factors. So blood contains nutrition and blood contains information. And the cellular signals come from the nervous system. So to understand how we create the blood composition, recognize this. We live in an environment external to our body as well as we have an environment internal in our body. And I say, well, how does the cell know what the environment has in it? And the answer is this. The function of the nervous system in the brain is to read the composition of the environment in which we are living. So the function of the brain is perception, what is going on in the world. And then the brain will translate that perception into signals to coordinate the body's response to that environment. So the signals from the brain are passed via the nervous system and the circulatory system to the cells of the body. These signals in the blood are the factors that are then controlling epigenetics, and so brain-mediated signals control behavior and gene activity by being transmitted through the nervous system and blood to the target cells. When we get to the level of the human, it's more than just the environment that is controlling behavior and genes. When we get to the human, we start to recognize that there is a mind. And when we put the mind into the scheme, it changes the flow of information for this reason. The brain picks up the signals from the environment, but the mind does an interpretation of those signals, and that interpretation is translated into the chemistry released by the brain. So the culture medium components controlling biology are not necessarily directly related to the real world. The culture medium contains information that is directly related to our perception of the world. If we have a misperception of what's going on in our environment, then you realize this. The signals that our cells receive internally is not the signal of what's really going on in the world. And so the cells don't see the real world. They only see our interpretation. That interpretation translated by the brain turned into chemicals that are released into the body, causing the composition of the culture medium that influences cells. So we now have to recognize that the environment that we are living in is really creating the chemistry that is controlling our biology. To simplify an understanding that, in this cartoon, what we can see is an image of a brain. And in one hand of that brain, there's a flask of culture medium. In the other hand, the brain is feeding that culture medium to cells in a dish. And I go, okay, that cartoon represents the fact is this. The brain is creating the blood, the culture medium, and that blood is then used to control the fate of the cells. So I say, okay, well, how does it work? And I go like this. When we have a perception, that perception is translated by the brain into the chemistry used in the culture medium. So I can say, when you open your eyes, let's say, and you see someone you love, the brain interprets love and translates it into chemistry such as providing the dopamine as a result of experiencing love, which is the pleasure we experience in love. In addition, when you're in love, the brain will release oxytocin into the culture medium. And the oxytocin is to bond you to that source of love. Then the other chemicals that come out of the brain include vasopressin. So when you're in love, vasopressin is a chemical that makes you more attractive. so that your partner, your lover, will stay with you. And lastly, brain activity in response to love releases growth hormone. And growth hormone, as the name says, is a chemical that supports the growth and health of the cells in your body. So a simple point is this: when you're in love, the chemistry released by the brain into the culture medium is chemistry that enhances your health, your vitality, and makes you much more attractive in the world in which we live. And this is a wonderful experience and a consequence directly due to the perception of love. But in contrast, if you open your eyes and you see something that scares you, there's a completely different set of chemicals released into the blood. No longer are those vital chemistries such as growth hormone and dopamine released when you see something in your environment that causes you to be afraid. Because in that situation, the cell will begin to release stress hormones and inflammatory agents that put your body into a protection mode to protect you from the threats on the outside. So if you put the chemistry of fear into the culture medium, it alters the expression of the cells. In fact, in a laboratory situation, if I create culture medium and put those stress chemicals into that culture medium and feed my cells, guess what? Within minutes, the cells start to get sick, and then shortly after, they begin to die, which really supports the known belief that stress can cause death. And in fact, stress is the most important factor that apparently is influencing the health on this planet. So we can come back and say, well, the function of the mind is to create coherence between what we believe and the reality that we experience. If you believe there's a threat, the signals of threat released into the blood will prepare the cells to engage in a protection response. But the problem is this: maybe there was no real threat. The cells don't know whether it's real or not because the cells only respond to the chemicals. They don't see the real environment. So all of a sudden I say, then if we believe we're under stress, we manifest stress in our physiology, even though the environment in which we live is not really promoting that. So all of a sudden we start to see that our thoughts, whether they're right or they're wrong, are actually changing our biology. Now, this idea of thoughts controlling our life is not new. 2,500 years ago, the Buddha said, what we think, we become. And in fact, that is exactly the story of epigenetics today. Epigenetics is saying what you're thinking is translated into chemistry, which will determine what you will become. So, 2,500 years later, Buddha is right, but now we know the mechanism. Signal plus protein equals behavior. Now I say, when the behavior is not supporting us, then we can say, we are expressing a dis-ease. Now, in this equation, I say, well, what can be responsible? What can cause a disease? The beautiful part about it is there are only two things on the other side of the equation. It's either the protein or the signal can be considered the cause of disease. So I say, well, yeah, proteins can cause disease because defective proteins lead to defective functions, which then are expressed as disease. So the question is, how much of our disease is actually controlled by defective proteins? And the most amazing fact is this, that flawed genes cause less than 1% of disease. So when we go back to our chart and I say, proteins can contribute to disease, and I say, well, they can contribute because defective proteins as a result of mutations or alterations of our genome then will mislead the fate of the cells, and in that case, cells out of harmony with the world express disease. Now we're down to a very simple understanding. If only 1% of disease is related to proteins, going back to the chart says, well, then the other cause of disease must be some alteration in the signal. So I say, the signal can cause up to 90% or more of disease that we experience on the planet. I say, well, how can the signal interfere with health? And I say, number one, if you experience trauma, let's say you're in an accident and you twist your spine and you interfere with the flow of information from the nervous system to the cells, then the alteration of that interference of communication could lead to a misunderstanding by the responding cells. So what's really going on, because the signals are altered. So trauma can cause disease by interfering with the propagation of the signals from the brain to the cells. Number two, if we put toxic elements into our body, such as the toxins that we acquire by eating industrial farm foods, toxic chemicals can engage in the signal pathway, but if they do, they don't promote a normal signal propagation. Toxic chemistry can distort the signal. So relevance is this. If the brain is sending a signal to control the cells and there's toxic chemistry in the pathway, then the signal that reaches the cells is by definition altered, and therefore the behavior of the altered cells can lead to disease. Thirdly, and perhaps the most important cause of disease influencing the signal, is really nothing wrong with the body at all. But what it represents is this: the influences of our thoughts. Our thoughts are what are translated into the chemistry. Now, why is this relevant? If we have a disease due to thought, it's not because the body is defective, it's because the signal is inappropriate. And this is really important because it is recognized now that our consciousness is the primary problem in the issues of health on this planet, especially as they relate to stress. And that this issue is nothing organically wrong with the body. It's just that we're sending inappropriate signals at an inappropriate time, which leads to inappropriate behavior, which we call disease. So I look over, I say, look, there are only three causes of disease that interfere with the signal. One, trauma, and trauma represents a physical impairment. Two, toxins, which represent a chemical impairment. And three, thought, which introduces an emotional impairment in the system. And collectively, all of these things result in what we've referred to as stress. So stress is the primary cause of illness, with proteins attributing only about 1% toward that pathology. And I say, well, is this really the understanding? I go, the American Psychological Association, in an article entitled Stress, How Does Stress Affect Us?, comes to the conclusion that from 75 to 90% of all doctor visits are actually due to the influence of stress. And all of a sudden I say, yeah, but then that means 75 to 90% of all doctor visits can be eliminated if we understood how to handle that stress. So I say, well, first of all, before you go deep into stress, we must recognize there are two kinds of stress. One is called eustress. Well, that's good stress. You say, there's a good stress? I go, yeah. If you play sports, for example, and you're out on the playing field and you're running and jumping and moving around, by definition, you're stressing the physiology of the body. But it's not a bad stress, because the resolution of this stress is the body's ability to strengthen itself, make itself be able to carry out those athletic needs. So yes, I'm stressing the body when I'm playing a sport, but it's not affecting me in a negative way. It's actually affecting me in a positive way, because it's inducing more health from my cells to accommodate my needs. The issue we really want to deal with is called distress. And the significance of that is a stress that promotes a negative environment. I say, well, what does distress mean? And it's very simple. Distress is a result of anything that gets in the way of your destination. Whether your destination is a career move, or whether your destination is a relationship, or whether your destination is trying to take care of your health or your financial background, anything that interferes with those, by definition, is a source of stress. So distress is the bad stress that interferes with health. And I say, well, how does it interfere with health? And number one is simply this. When you put cells in a petri dish, I can create an experiment where I create two sets of dishes. In one set of dishes, I put nutrients in front of the cells to support their growth. In the second set of dishes, I put a toxic element that threatens the cell's life. I put these culture dishes back into the incubator, wait for a while, come back, take the dishes out, and then I look, where are the cells in each of these two dishes? Well, number one, when you put nutrients that support the life of the cell in the dish, the cells move to the nutrients to take in the nutrients, assimilate them, and put them in their body. But in the Petri dish with the toxic elements, the cells don't move to the toxic element. In fact, they move the opposite way. The cells move away from the signal, and in this case, in the form of protection, they also close themselves down to wall off the threat. So we have two different behaviors based on the signals. A positive signal causes the cell to move to the signal, open its arms, and take in, assimilate the positive signal. A negative signal causes cells to move backwards away from the signal and close themselves down in protection. The point is this: cells cannot move forwards and backwards at the same time. Cells cannot be open and closed at the same time. The net result is a very simple conclusion. Cells can either be in growth, moving to the signals and assimilating, or cells can be in protection, moving away from the signal and walling themselves off. And the important part about this is that these behaviors are mutually exclusive. A cell cannot be in growth and protection at the same time. So, when we look at this picture and understand it, then what we can recognize is a simple fact. Protection mechanisms wall off the body and stop growth. I go, well, that sounds like a very bad situation. Why would we do that? Why would we shut down our growth mechanisms when we're under stress? That might be the time we need to activate those mechanisms. The answer is no, for a very simple reason. Historically, in evolution, go back to primitive people. And I say, well, tell me about their lives. And I go, well, very simple lives, looking for food, hanging out in the trees, and just enjoying the Garden of Eden at the time. But then I say, occasionally, a saber-toothed tiger shows up. And I go, well, that means the human has to protect itself. And I say, well, what's the human going to do? Engage in fight or flight. This is the engagement offered by the hormones of the adrenal system. I say, yeah, but what does that mean? I say, well, imagine you're running away from a saber-toothed tiger, and I say, how much energy do you want to allocate to your fight or flight mechanism to escape the tiger? And the point is logically very simple. All the energy that I can make available, I want to have available to escape the tiger. So I say, yeah, but the energy in the body is being used to do many different things, such as growth and maintenance of the body, taking care of it, growth functions, and energy is actually used for the immune system. And it uses a lot of energy, that immune system, only because, as you well know, when you're sick, most people don't have enough energy to even get out of bed. So the immune system uses a lot of energy, and growth uses energy. So I say, you're being chased by a saber-toothed tiger, how would you split up the energy? And I would say, stop the energy for the growth mechanism. Why? I don't need to grow right now. I need to escape and save my life. I don't escape. Growth is irrelevant. The same thing applies to the immune system. If I'm having a bacterial infection and I'm being chased by a saber-toothed tiger, what would be the benefit of funding the immune system with energy? Because if the tiger catches you, that bacterial infection is not your problem anymore. It might even be the problem of the tiger. So the point about it is simply this. In a state of threat, stress hormones are released from the pituitary master gland to control the protection response in our body. The stress hormones do two things. They shut down all mechanisms that utilize energy that are not necessary from escaping from the tiger. So growth and the immune system are inhibited under the influence of stress hormones. When the system evolved 1 million years ago, the only stresses that people had were that occasional saber-toothed tiger. So there will only be short intervals of using a protection response, just long enough to escape from the tiger. When the individual had escaped the tiger, they were no longer needing a protection response. That stress system would shut down and the system would return back to normal growth. But that was 1 million years ago. Today, the stress that we face in our world is not little short bursts, but actually 24/7, 365. Every day, based on what's going on in the world, financially, health-wise, whatever, stress of that information is released in our body in the form of stress hormones, and they end up shutting down the growth and the immune system. And I go, well, That's not the original intention, a short burst of protection, because chronic protection leads to chronic problems because of its ability to shut down the maintenance of the body and the immune system. So all of a sudden you realize, why do we have so much illness in regard to the world we live in? And the answer is not because of our genetics and our biology, but because of the level of stress that the citizens have to deal with from day-to-day life. And that if we could eliminate that stress, then what we can do is actually recover our health again. So our health is not due to a defect in our body. Our health is just due to a mechanism that is out of control because of the amount of stress that we are creating in the world. So, two consequences of stress. A, when the stress hormones are released into the body, they start to close the blood vessels in the gut, and that stops the flow of blood to the gut. The blood, of course, is the source of energy. And I say, well, why would I stop the flow of the blood vessels in the gut when I'm being chased by a tiger? And the answer is simple. If you close down these blood vessels, it pushes the blood to the arms and legs, which are going to be used in the escape process. So we're just allocating energy to the somatic system to escape the threat. But if the threat is prolonged, then you have to realize those blood vessels shutting down the gut functions will actually inhibit the maintenance and caretaking of the body, and prolonged stress will have detrimental effects on our fate in regard to our health and our ability to survive. So that's aspect number one, shutting down the growth. Aspect number two I also mentioned was this. The immune system uses a tremendous amount of energy. So the idea of an internal immune problem and an external saber-toothed tiger easily says, forget the immune system at this moment. Let's put the energy into running away from the tiger. And I say, yes, that is one of the functions of stress hormones. Stress hormones shut down the function of the immune system. It is so effective that medical doctors give patients that are going to receive a transplant of an organ, they give those patients stress hormones before the transplant. This reason is quite simple. The ability of the stress hormones to inhibit the immune system that actually protects the recipient of the new organ from having their own immune system reject the graft. And this supports the life of the transplant. So the effectiveness of inhibiting the immune system is so much, it becomes a therapeutic tool in organ transplant. So I say, okay, our life is now under threat, and our hormones in our body are shutting down growth, and the immune system, there's only one consequence of that, and that is a breakdown of the system and infection. And this is really the primary cause of the illnesses that we have on this planet. And one other aspect, which I call adding insult to injury about stress, is this. The prefrontal cortex portion of the brain, which is just behind our forehead, is the site of our conscious mind. And interestingly enough, while the conscious mind is creative, it's a very slow processor. The hindbrain has reflex behaviors, which are instantaneously fast responses. When they're under stress, the adrenal hormones squeeze the blood vessels in the forebrain just as they squeeze the blood vessels in the gut. I say, yeah, but when you squeeze the blood vessels in the forebrain, it forces the blood to the hindbrain. That, in other words, the blood energy is not being used in the forebrain under stress, but is being used to engage the reflex behavior of the hindbrain, the much more effective mechanism to save us in a very scary, stress-inducing situation. And I go, relevance is this. Forebrain is consciousness, hindbrain is reflex behavior. And when we're under stress, the hormones shut down our consciousness and cause us to behave in a reactive, reflexive mode. That makes us less intelligent. The net result is there are three consequences to elevated stress hormones. One, shut down the growth using blood vessel closure in the gut to stop the functions. Two, shut down the immune system because stress hormones inhibit the inflammatory role of the immune system. And three, stress hormones make us less intelligent because it shuts down consciousness and engages reflex behavior. You put all these three elements together, and then all of a sudden you have a profound understanding of the cause of disease. The two basic ones, physically, not supporting the growth and maintenance of the system, and inhibiting the immune system, lead to a physical breakdown of our biology. And that is why up to 90% of doctor visits are primarily due to stress. In addition to the immediate influence of stress, stress has a long-term interference with our vitality and the survival of our biological system. And it influences this effect via something called telomeres. And you say, well, what are telomeres? And I say, understand that a DNA molecule is a double helix, as Watson and Crick revealed. That it will stay functional as long as the molecule stays in its helical configuration. But if you start to unfold the double helix into single strands, this opens the DNA to a breakdown process where you can destroy the gene programs. So you don't want the helix to unfold. I said, well, how do you prevent it from unfolding? Well, to give you an idea, I'll show you a picture of a shoelace. And at the end of the shoelace, there's a special little plastic cap that holds the threads of the shoelace together. It's called an aglet. And I say, well, great. The aglet holds the filaments of the shoelace together and makes it easier for us to stick the shoelace through the little holes into the shoe so we can tie our shoes. I go, yeah, but if the aglet comes off, the end of the shoelace frays, and then now you start to ruin the function of that shoelace because you can't get it through that little tiny hole anymore. And I go, in a parallel understanding, the DNA is the wound up version of the double helix, and to prevent it from unfolding, there's the equivalent of an aglet called a telomere. That holds the ends of the DNA together to prevent them from unfolding, okay? If the telomere is gone or breaks down, then the DNA will start to unfold, and that would have a negative aspect on our health because it can cause many of the diseases that we experience, such as aging, depression, and other physiological things like cancer, etc. So the telomeres are playing a very important role. And yet what we've looked at is that they appear to be nothing more than the equivalent of an aglet to hold the DNA together. I go, no, there's more to the telomere than that. And that is this. Why have the telomere? Yes, to hold the DNA double helix together, but there's another more important reason. These telomeres, as you can see in the picture of an isolated X and Y chromosome, these telomeres stained red are at the end of the DNA where we would expect to find them. And I go, but what do they represent? They're extensions of the DNA, but they don't program anything. So they're not genes, they're just nonsensical in one aspect, extensions of DNA, no gene program. I say, so yes, they're there to hold the DNA double helix together. And I go, yeah, but there's another important reason for telomeres, and that is this. In copying the DNA, you have to split the DNA apart into single strands. And when you do that, an enzyme called a DNA polymerase will land on a single strand of DNA, and like a train going down the tracks, as the polymerase enzyme moves down the length of the DNA, behind in its wake, it leaves a copy of the DNA. So imagine a train going down the track, and right behind the train, there's a reproduction of the track. So all of a sudden, now I have two tracks where I had one. This is how you copy the DNA. And I go, well, this is really great, but here's where the problem comes in. When that enzyme reaches the end of the DNA, and it's sitting on the DNA before it falls off, it cannot copy the piece of DNA upon which it is sitting. So I create a copy of the DNA that I'm on with the enzyme, but when the enzyme gets to the end of the DNA strand, it cannot copy that last piece. And I say, well, what's the consequence? I say, the new copy of the DNA is shorter than the original copy. I go, oh, okay. I say, yeah, but then the next time it divides, the same thing will occur. So then the next copy of that DNA is still going to be shorter than the previous copy. Every time you copy the DNA, you lose a piece of the telomere because it cannot be copied by the enzyme because the enzyme is sitting on top of it and needs to pass over it. And if it can't pass over it, it can't leave that DNA behind. So the net result is this. Telomeres, yes, provide for holding the DNA together to prevent it from fraying and then causing its breakdown. But as, or more importantly, the telomere provides a platform that allows a large number of reproductions of the DNA. Considering that every reproduction is shorter than the previous version, it will say what? At some point after so many cell divisions, the DNA called the telomere will be lost because it's shortened every time previously. And I said, well, what happens if you copy the DNA and the telomere's not there? And I say, oh, well, the next copy is going to be short again. But what's important is what you're shorting is not an extra piece of DNA. You're shorting a piece of a gene program that makes a protein vital to our biology. So when the telomere runs out, the next copying of the DNA will interfere with the genetic code and alter the expression of the organism. It was then understood that telomeres determine longevity. As long as you have telomere, you can reproduce the DNA, but if you divide the cell too frequently, the telomere disappears, and the next version, shorter version, will be shorter because you're cutting off a portion of a gene program. And that interferes with our biology and can cause aging, disease, and depression. So I say, when this was originally understood, a scientist by the name of Leonard Hayflick in the 1960s said, well, if every division makes it shorter, then there's only a finite number of cell divisions a cell can have. When it reaches that number of divisions, after that, disease will result, and then death will occur. So it said, our life, the length of our life, is determined by the length of the telomere. The shorter the telomere, the closer you are to aging yourself out of existence, because when the telomere is gone, that's when the disease enters into the picture. So the original thing said, all humans have a fixed lifespan because they have a fixed DNA extension called a telomere. And that all of a sudden led to counting how many cell divisions a cell will be able to experience before losing its telomere, and then put that into context of how many cell divisions we experience in our life. And it says, well, a human may have a life of anywhere from 80 to 90 to 100 years based on the amount of telomeres that are maintained in their body. Well, that said, oh my God, our lifetime is fixed. However, a woman by the name of Blackburn identified an enzyme called telomerase. Telomerase is an enzyme that extends the telomeres. I said, oh my goodness, if there's an enzyme that extends the telomeres, then the concept of a fixed life is totally wrong. Because as long as I can extend the telomeres, I can continue to divide the cells and maintain my life. So there is no limitation on how old we are, because we can use the extended telomeres to extend our lives. This then leaves us to a final understanding about the influence of telomeres in our lives. And it really comes down to this aspect, and it says this. There's the enzyme that can influence the production of more telomeres. If we activate the enzyme, we can extend our life and our health. If we don't activate the enzyme, our life will be shorter and will be influenced or affected by disease much more easily. So I say, okay, there are things that will enhance the telomerase, which will increase our life and our health, and there are factors in our life that can stop the telomerase and in the end cause us to have premature death and disease along with that. So I say, well, what are the conditions that are responsible for, let's say, inhibiting the telomerase that cause us to have the diseases that we have on this planet? And the answer to that is, here is a list of the following factors that will stop the telomerase activity, shorten your life, and cause you to have more disease. And these factors include stressful prenatal development. And the significance of that is, if you're stressing the cells in the very early part of life, you're promoting more cell divisions very early that will then use up the telomeres, and because of the stresses, you're not going to extend them. So the result is stressful prenatal development will shorten the duration of an individual's life. Childhood abuse will do the same thing. Now, people think childhood abuse is physical abuse, hitting your child. And I go, well, that is childhood abuse, but what you must understand is this: childhood abuse can also result from verbal abuse. So they say, no, I didn't hit my child, I just yelled at him. I say, no difference. Yelling at the child, hitting the child have the exact same effect on the telomeres because they inhibit the telomerase activity. Domestic violence, another factor that will shut down the enzyme. Post-traumatic stress disorder is also in that community of inhibitors of telomerase. Two last things to consider are this: nutritional deficiency. And the reason why that interferes with the telomerase is that we don't have the metabolic support to provide for telomerase activity. And let me just add this as what I think is the most important of the influences that shut down telomerase and lead us to disease, and that is a loss or lack of love. Love feeds the system, encourages health and growth. As I said, the experience of love releases growth hormone into the body, and in that environment we can create more health. But when there's no love, the telomerase activity will shorten itself. All of these things between violence, stress, lack of love say that we're not enjoying our lives. And the response to that is then our biology says, oh, okay, well then we can shorten it right away because we can use up the telomerase and get out of this lousy situation. I say, yeah, but look at the opposite consequences, the opposite things that result in enhancing the telomerase activity. Number one, good nutrition, and especially including omegas. Omega lipids are vital to supporting telomerase activity. Exercise, as I said, that's eustress. That's the thing that builds up the biology, and in building up the biology, again, that's another activator of telomerase. Love and self-love increase health and are feedback to the system. It says, I love my life. Just literally what you're telling yourselves is, I want more life. Love enhances telomerase to give us a greater opportunity to experience love on this planet. Along with love is positive outlook. If you see the future is bright, you're telling yourselves, I want to be here for the future. If you have a very negative outlook, what you're telling yourselves is, I'm afraid to be here and I don't want to be here because it's going to be very stressful, whatever it is, and you'll shut down the enzyme. So love, positive outlook, happiness and gratitude are all thoughts of the brain translated into chemistry to engage telomerase, because those are the thoughts that say, I love my existence on this planet, I want to be healthy and I want to maintain myself, and as a result, these thoughts and actions cause the telomerase activity to be increased, which ends up extending our life. And lastly, and most importantly, is that being in service activates telomerase. What does that mean? Are you a caregiver? Are you providing health for somebody? Are you providing something that other people need to stay alive? You start to get a conscious understanding that says, I am responsible for taking care of these things. I don't have the time to die early. I need to stay healthy longer because I have these jobs. So being in service is actually promoting telomerase activity and increasing your lifespan. Okay? And let's consider the opposite, which usually affects people after they retire. I'm retired. I have nothing left to do. What does the feedback of that thought have to do with our biology? Simple. When we have no function or reason to be here, we inhibit the telomerase because the telomerase was extending our life, and our thoughts say, we don't need any more extension. We're finished. I'm retired. I have nothing left to do. And I say, just that thought alone can stop the telomerase activity, and that result causes us to shorten our lifespan and engage in more sickness as well, because the loss of telomeres are one of the factors that increase the problems in our biological system. The summary is this: our thoughts are translated into chemistry that shape our body. The shape of our body is complement to the thoughts. Positive thoughts give us a positive, healthy biology. Negative thoughts can actually introduce any disease and death. That these thoughts control our biology on a day-to-day basis and interfere with our metabolic and physiological systems if they're not supporting us. And then I go, but these thoughts and behaviors also influence our longevity, because these thoughts can alter the activity of telomerase enzymes. If we stop the activity of the enzyme, we are shortening our lifespan and increasing the chances that we will have disease. In contrast, if we engage in those behaviors, such as positive thinking, being in service, being in love, those are things that engage the telomerase enzymes, and these activities then prolong our life and make us healthier. Conclusion is very simple. We're not victims of our genetics and our heredity. Epigenetics reveals how these thoughts that we've been talking about influence the genetics and the behavior of our cells. Therefore, what we have to understand, and what we must also help others with, is that our health is not out of our control. Our health is directly under our control. The control of our perceptions, our beliefs, and attitudes actually is what will control the stresses. And the stresses are the things that cause the disease. If you stop stress, you can stay in health. And as a mission for healers around the world is to get the stresses out of the patient. So once the stress is removed, the system is able to rebuild itself using the chemistry of the brain to support growth and increasing the activity of telomerase activity, which supports a healthier, long-living biological system. The conclusions for me are important because if you understand them, you can control your life and you can help others control their lives. And so this wonderful new information about stress and telomeres is fundamentally important for healing our planet. Thank you for your attention in this interesting topic.
Quantum University is the world's largest institution of higher learning to provide online degrees and certification programs in holistic, alternative, natural, and integrative medicine based on the science of quantum physics. The International Quantum University for Integrative Medicine, IQUIM, also known as Quantum University, is an online degree-granting institution located in the state of Hawaii in downtown Honolulu. Students can earn bachelor's, master's, doctorate and PhD degrees, leading to professional board certifications. Quantum University's mission is to build a new future for medicine, one student at a time. Graduates of Quantum University take their health and wellness career to the next level and follow their dreams to start a new, exciting, successful and meaningful career in integrative medicine.